go over your introduction. So Dr. Sheehy is an acclaimed entrepreneur and health visionary with over 15 years of technical expertise in optical and ophthalmic engineering. As the founder and the CEO of C2SHiP Technologies, Christy leads a team of renowned scientists, engineers, and business strategists on a mission to innovate AI-driven optical solutions that enable proactive healthcare. In May 2023, C2SHiP received FDA 510K clearance on their device, the Retinatrack, a revolutionary retinal eye tracker that enables quantification of oculomotor function. Dr. Sheehy spun this technology out of her PhD work from UC Berkeley optometry and holds an exclusive license on its use in healthcare. In addition, Christy has aspired C2SHiP placement across several startup incubators and accelerators, including Berkeley Skydeck, Stanford's StartX, NIH C3I program, and the NASDAQ Leader of Tomorrow program. She has received over $8 million in funding from venture capital funds, angel investors, and NIH grants to accelerate her team's vision of developing eye tracking solutions that promote longer, healthier lives. Dr. Sheehy, please share your screen and take it away. Great to have you here.
Awesome. So I actually have shared my screen. Can you guys see that?
Yes, we can see your screen.
Perfect. All right. Hi, everyone.
And by the way, your screen looks great.
Perfect. Hi, everyone. I'm Christy Sheehy. I'm the CEO and co-founder of C2SHiP Technologies. I want to thank UND and their faculty there for inviting me here today. I am in a hotel, so my Wi-Fi is a little bit sporadic, so I'm going to leave my video off, but please, if at any time my signal is crummy, just feel free to interrupt me and I can re-say anything that might have been missed. So I'll start off with a quick disclosure. Again, I'm founded in C2SHiP Technologies and the core technology I'll talk about today. So I just want to get that out in the open. So I'm going to dive right in. Many of us think about our eyes moving, but we don't necessarily think about what those movements mean. So there's 4 main types of movements that the human body can make. Saccades, if you've ever been to the doctor and they say, look at my finger, look at my nose, that type of movement back and forth between finger and nose would be a saccadic eye movement. There's also smooth pursuits. So if a doctor ever said to you, follow my finger, that would be a smooth pursuit movement, steady motion over time. And then vergence would be if a doctor held their finger close to your eyes and then moved it far away. So looking at things that are near and far and kind of how your eyes will turn in or turn out accordingly. And then there's fixational eye motion, which is the eye motion that doesn't get talked about too much because it's often not able to be measured. And that's actually going to be the focus of my talk here today, looking at the motion that happens while you try to keep your eye as stable as possible. So one of the beauty of having fixational eye motion is that it allows us to have a constant brain refresh. So if our eyes were not always in motion, the outside world would actually fade away from view in about 80 milliseconds. And so there's a little fun exercise you guys can do here at home. If you look at the center of this circle here on the right, so there's a blue circle with a white dot in the middle. If you keep your eye as still as possible and focus on that white dot, you'll start to notice that the outside circle has subtly faded away from view. And so this is the perfect example of your neurons adapting to a scene and no longer thinking it's viable information, and therefore it fades from your peripheral view. So it's kind of that perfect example of if our eye isn't moving, we might not be able to capture the outside world. And this is called scientifically the Troxler effect. So imagine if your eyes don't move enough, the visual world could fade away. In contrast, some individuals might actually have eye motion that moves too much. So over here on the right is a picture of tacos, which makes me ready for lunch already. But you can see that the tacos are very subtly pulsing or moving. And so there are types of eye movements where if I'm fixating and I'm having intrusions or some sort of abnormality, the world might actually appear to be moving even though the world is stationary. So I always kind of joke that fixation is like the Goldilocks of eye movements. You can't move too much because the outside world will start to kind of jitter and shake. But you can't be too stable because then the outside world would just fade away from view. So with fixational eye motion, there's really this ability to see very subtle changes. So clinically, if you were to be at a doctor's office and they were to look at bedside, your eyes should look relatively static and stable. There shouldn't be any big intrusions. You should be able to stare straight ahead. But if you then zoom into the very back of your eye and look at the back layer of your retina, or look at the back layer of your eye called the retina, you'd actually see that there's really subtle movements that are happening over the course of time. And some of these motions can be as small as half the size of a human hair. And so again, these movements will allow our brain to have a constant refresh of the world. And there are three main types of fixational eye motion. One of them is drift. So the image that you see here on the right actually would represent your cone photoreceptors at the back of your eye. They're what help us see color. So they're painted here in red, green, and blue to represent the wavelengths with which they function at. And you can see that white dot or white line kind of traversing over the photoreceptor mosaic. So for drift, this is going to be kind of the arching, slow moving, low velocity movements of the eye during fixation. For microsaccades, these are more of the quick ballistic movements that are happening, and they're going to traverse, hundreds of photoreceptors as opposed to drift, which might only traverse maybe dozens. And then we have a third type of eye movement called tremor. And this is typically thought of a high frequency superposition on your drift of around 80 hertz. So generally for fixation, these are the three eye motions that would come up in literature. And again, they help our brain to have a constant neural refresh for the outside world. Now, the beauty of the visual system is that the brain is extremely active in how we view and see the world. So there's two kinds of ways in which we view the visual pathway. The afferent visual pathway can kind of be thought of as light going in and getting processed. So if I'm looking at the outside world, I have light coming into my retina. It's going to go to my ganglion cells, which then go down my optic nerve, the chiasm, the track, all the way back into the LGN and visual cortex. So that's kind of how I see the world from the outside going in. And there's ways in the clinic right now that you can capture this visual system's performance, so to speak. Optical coherence tomography, contrast sensitivity, color vision. Those are tests that can really assess how well this pathway is working. But the pathway I'm going to talk about today is the efferent pathway. And this is the pathway that we need to move our eyes in response to a stimulus in the outside world. And with the efferent pathway, what's really cool is it actually innervates all four lobes of your brain. So to literally make an eye movement, we have to use every single lobe. So this picture over here on the right, the green would be the excitatory pathway, the red, the inhibitory pathway. And so you have excitatory signals from cortical regions like your frontal eye fields, secondary eye fields, parietal eye fields. And then this information can then travel from the superior colliculus down into the brainstem, talk to our cranial nerves, and then ultimately our extraocular muscles to help us actually move our eyes. And this is where, again, eye movements would be made from the efferent pathway. So I know that was kind of a crash course in how the brain and eye is related, but it's a beautiful way to show just how powerful our brain is working every day, literally just to make our eyes move from one target to another. So the brain is constantly being taxed just to move our eyes as we look around. And scientific literature has shown that eye movement has been able to be documented as changing in a multitude of conditions. So eye movement literature, of course, dates back decades. But in the space, saccadic eye movement, smooth pursuits, vergence, fixation, they have all been able to be provided, or they have all been able to be documented in each one of these conditions. And I always say the beauty of what we do here at C-Light, coming as a scientist, I'm very active in the research space. And so I love to be able to collaborate with clinicians potentially on this call, companies on this call, to dive deeper into some of these areas to look at fixation, because many of them have really only focused on the larger kind of gross motor movements, looking at saccades, smooth pursuit, and vergence, and might not have done as much in the fixational space. So again, this list is not exhaustive. There's definitely more conditions that could be impacted with eye movements of the eye, but I just wanted to give the team here an idea of just how many touch points we have: neurodegeneration, neurologic, retinal, acquired brain injury, psychiatric, eye motion changes in elite sports performance. It can even change for cardiac conditions as well. So based on this, I'm going to dive into a little bit of the work that we're doing here at C-Light on the research side. Again, our technology is approved as an eye movement monitor, but we work with fantastic university researchers that have helped us to really start to understand that eye-brain connection and be able to build algorithms and software to start to look more deeply at these different conditions. So a lot of the work I'll talk about today actually came out of my postdoc at UCSF. And so during my postdoc, I brought the technology that I built during my PhD into the clinical world. So a tracking scanning laser ophthalmoscope is the technology that I'll be talking about today. Over here on the left, people always joke, it's kind of like, optics IKEA, where you're basically setting up a series of mirrors that relay the light from our laser to be scanned onto the back of the eye or the retina. So 840 nanometer light would enter into the system. It would then be passed through a series of telescopes as it relays on to two different scanners, and then finally to the back of the eye. And a person would be putting their head right here, having their temples stabilized so that their head motion was limited. And that's when we can start to see these very subtle movements of the eye. So what you see here in black are vasculature. Each little white dot here is a cone photoreceptor, or what we use to see color. And then some of the videos I'll show you, if you look inside the capillary networks, you'll actually see some subtle blood flow as well. So just a really exciting real-time video of the eye in action. And so this technology we've now spun into a commercialized product called the Retinatrack. It is a 510(k) cleared retinal eye movement monitor. And some of our early customer base I list down here as some of the fantastic universities that we've worked with for NIH grants and for other foundational funding as well. So how we actually do eye tracking, I find very exciting. And I like to think of it, for those of you familiar with cameras, I think of it like a rolling shutter. So with our particular technology, we raster scan the eye pixel by pixel. And what that means is what happens and is recorded at the top of the frame might be different than what happens at the bottom. And so because of that, you can think of it like a rolling shutter effect, and you can split a single frame into multiple strips and then cross-correlate or match those strips to a reference. And so what ends up happening when you do that is you get a really high fidelity readout of horizontal and vertical movements over time. And in the past, within the clinical community, these eye movements were used just to create a better structural picture and then ultimately thrown out. But what I wanted to do and really focus on for our work is harnessing those eye movements that usually would just be used to stabilize an image and actually use these as the functional output of the system that we sell. So we're looking at translational movements, both horizontally and vertically over time. And a lot of people will ask and kind of be curious, well, Christy, when should I use a retinal tracker versus a pupil tracker? When does that really matter, or does it matter at all? And so some of the work that we had done early on in our company is just understanding that exact question. So you'll notice here on the left, I have a picture of a pupil, an iris, the sclera, and we're tracking kind of those different structural modalities on the outside of the eye. One thing you'll notice is that this pupil, or the hole really that brings light into the eye, it's rather dynamic. So the pupil itself is going to be constricting, it's going to be opening, and so it's kind of like a constantly moving target. When you go to the back of the eye, or the photoreceptor layer, that's going to be really kind of static structure to look at. And so when you're looking at photoreceptors and how they move, you can lock on to individual photoreceptors and then watch how those are moving over time, because they're not going to be dynamically changing, on the millisecond to second scale. And so what we found is that the retina is really fantastic for looking at very subtle movements. And then the pupil is going to be really great for your large gross motor movements. So the look at my finger, look at my nose, follow my finger, those are going to be really great to look at the pupil. And then fixation is kind of the sweet spot for retina. And I apologize if these videos have any lag or delay due to my Wi-Fi. I'm hoping that they show up cool, but I'm happy to share this presentation after to anyone who wants to see the videos better. And so what we found is that once you get under about 2 degrees for the pupil, that's when you start to lose a little bit of the fidelity of the tracking. And so, 1.5 degree here, you're looking at close matching. You have green for pupil, you have yellow for retina. And then once you move to about 1 degree, you start to see that amplitude kind of differing even more. But really, once you hit that 0.5 degree mark, that's going to be where pupil is basically just going to be noise, and where the retina is really going to shine. And the beauty of that is that fixation on average is 0.5 degree or less. So we're looking at visualizing movement that may have already previously just been completely invisible to existing technologies, and definitely not seen at bedside. And so that really brings the idea of bringing an entirely new stream of healthcare data into the world, and being able to visualize subtle movements that may not have had a voice before. And so here is a real-time video kind of some of the recordings that we've gotten from various research studies. On the left here is a healthy control eye. The movements there are kind of minimal. There's very subtle drifts. There's a microsaccade happening in the middle. The individual is relatively able to hold their gaze. And then within our study, we found some abnormalities. So in the center here, we have what's called a square wave jerk abnormality. So the person's trying to fixate, but their eye just keeps bouncing back and forth. This can become really problematic. This might be like the taco example, where if your eye is involuntarily moving when you're trying to keep it still at a large rate, this person actually complained that the outside world was jittering and shimmering, and it got worse with. So this is when you can really zoom in and see these movements closely. And the cool part is even though the movements might look large, they're actually only about 100 to 150 microns. So very, very subtle changes. And then here on the right is a subclinical nystagmus. So nystagmus happens when the eye basically is drifting away from the target, and then it snaps back. So for the engineers in the house, it's kind of like a slow phase, fast phase match. And then the square wave jerk for the engineers in the house kind of looks like a rect function. The eye snaps back, there's a little refractory period, and then it corrects it. So that's when you can start to see how eye motion actually becomes patternistic, and could be a beautiful opportunity for AI and machine learning to help us really start to segregate out those different patterns. And so some of the research I'll present today, again, this is research. It's not an indication for use for the device by any means, but it's just some of the studies that we've done in the research realm that have shown changes in fixational eye motion. So I'll talk a little bit about different disease cases that we've looked at with collaborative partners at universities, all the way through elite performance. So some of the early work I had done during my postdoc with the TSLO device, the research grade machine, was looking at how changes in multiple sclerosis progression might impact eye motion. So We had about just over 100 patients with MS and 100 controls. Their average ages were about the same, just under 50. And MS tends to happen at a 3-to-1 roughly ratio, 2 to 3 more likely to be women. And then in general, most cases are going to be of the relapsing nature, where an individual might have a sudden kind of burst of disease activity, and then that disease activity lessens. But there is a kind of a 30%-ish occurrence in the population of the progressive form of the disease, where you're just continuously getting worse and worse over time. And then RIS is, it means radiologically isolated syndrome. So they haven't been diagnosed with MS yet, but their MRI showed something that could be it. And then disease duration. So we had the luxury of tacking on to a longitudinal study at UCSF called EPIC, where they've monitored MS patients over the course of 15 years. So some of the patients have really been along this journey on average for 15 years of disease duration, all the way, of course, from someone just diagnosed through 45 years. And then for EDSS, what that is the disability level of a patient. So it's a way in which MS clinicians can kind of look at 7 different systems of the body and determine an overall disability score for that patient. And it's from a scale from 0 to 10. 0 means I have MS, but I could run a marathon. And then 10, of course, would be death by MS. So it's kind of a disability scale throughout there. Many individuals are on disease-modifying therapies, so we do note that in our records as well. And so what we wanted to look at was really how changes in microsaccades, or those quick jerk-like movements during fixation, how they might change with disease. And so when we think about what eye motion looks like from a time series standpoint, I have that plotted here. So blue is the vertical movement, red is the horizontal movement, and you can plot these over time so that you can begin to look for patterns or abnormalities. And so these right here are actually square waves. So these little rect functions where the eye jerks one direction, the brain says, wait a minute, and then tries to correct itself. So once you have these recurring kind of back to back to back, that's when a patient might have perceptual consequences. You can also look at eye motion from an angular extent. So if I plot this with a polar plot, I can kind of see the amplitude and direction of these movements as represented by little red circles. So you'll see square wave jerks are mostly in the horizontal direction for this particular example. And one of the cool things that the human eye does, which actually relates to astronomy, is this used to be thought of peak velocity versus amplitude or magnitude for stars. But actually the visual system follows this very same relationship. So as you increase peak velocity, you also increase amplitude. So imagine the more I have to move my eye, the more pedal to the metal I'm going to need to get there, the more velocity I'm going to need to make that bigger movement. So it extends from the fixational level all the way through to that voluntary saccadic level. So fixation, again, trying to keep my eye still, but I'm making microsaccades. And then the active voluntary movement of look at my finger, look at my nose. So these all fall all along that same linear relationship of the main sequence. So these are areas with which we can study outside of the different metrics that I listed here for amplitude, frequency, speed, and directionality. And so looking at all of these different metrics, we were able to actually show how microsaccades correlated with disease progression as represented by that EDSS score, the disability score. So down here, I show the disability scale. I have a few animations just to help you understand kind of what it means. So here, 0 to 1, I'm still running. You can barely tell I have MS. Around a 3 to 3.5, I can't run anymore. I'm kind of walking. When you get around 4 and 5, you might need assisted walking aids. And then 7 and 8, I'm usually in a wheelchair. And so what we found is that as the disease progressed over time, eye motion metrics would actually correlate with that disease progression. And so instead of having to wait for changes or large gross motor movement abnormalities with ambulation, you could actually non-invasively look at the eye and start to look at motor function on a really fine scale and be able to see subtle changes as well. So we noticed the number of microsaccades correlated with a cognitive test called the Simple Digit Modalities Test. It correlated with a 9-hole peg test, which is a test of hand dexterity, putting pegs into holes. We noticed that microsaccadic amplitude correlated with fatigue, as well as microsaccadic velocity. And when we compared that to controls, there was no correlation, which means the fatigue was really disease-mediated fatigue from MS, which was really exciting for us because it showed us that it's not that if you're just tired, you might have a change in eye motion. The fatigue is really that disease-mediated fatigue. And so some of the work that we've been doing here internally at our company for research is kind of understanding how can we build AI pipeline? How can we start to use eye motion potentially as a future biomarker, future use case? And so again, with the research study, the TSLO that we were working at during my postdoc, we utilized that data to start to feed into machine learning algorithms. So looking at the low disability patients, the patients that don't need assistive walking, and then comparing them to age-matched controls. And we gathered over 250 patients during the time of my postdoc, and we were able to actually differentiate yes-no disease with a 92% area under the curve. So for us, this was really exciting because it showed for the first time that fixation, the involuntary movement that happens when you try to keep your eyes still, it has the potential to actually show differentiation between sick versus not. And how we thought that was exciting as well is these features that could help to differentiate can also be tracked over time. So they can be dug into more deeply with information like the previous slide where you're looking at, changes as the disease progresses. And some of these results here are actually published in papers from my postdoc back in 2020 in the Journal of Multiple Sclerosis. We've also had some additional funding from the ADDF Alzheimer's Drug Discovery Foundation, which is backed by Gates Ventures. And we started to look at how eye motion might change in the earliest stages of mild cognitive impairment. And so we had a small cohort here, kind of just a pilot study that we ran with a split of male versus female. The average age in both the control and MCI group is around 72, 73. And we wanted to look at how, again, fixational metrics might differ between the two groups. And so here is where it gets kind of scientifically exciting, is that if you just give individuals kind of a typical target to look at, a crosshair, there wasn't really all that much differentiation. But once you started to change up the target, and here we call it center versus corner. So once you started to change the target that they were looking at, you could actually begin to differentiate the features between MCI and control. And so this for us was a validation that for neurodegenerative disease, future research would be really exciting, looking at what types of target might elicit the most instability or changes in a repeatable way for that population. So you can kind of think, if I'm going to have a disease case where attention matters, how might distractors to a target make a difference? If I'm going to look at individuals who have other neurodegenerative conditions, maybe crowding would make a difference. So again, this is really where some of the fun university collaborations can happen, starting to think about different ways in which we look at targets and how they relate to disease changes. We also collaborated with the University of Pittsburgh, their team over there for concussion, absolutely fantastic group and individuals looking at concussion recovery. And so for this, there is a paper published back in 2023. There's also an earlier paper published by Leonard et al. Before this, just looking at concussion versus control. But what we found is that individuals that had fixational abnormalities in the earliest stage, more of the acute stage of concussion within the first 21 days, those individuals were more likely to have a prolonged recovery. So that was the first time in the research world that we could show fixation could be predictive of a recovery process. So if I have fixation in my acute stage abnormality fixation, I might have a longer haul of my recovery. And this was an AUC of 81%. So again, looking to expand this study. We have a phase 2 grant now for just over $2 million with this group. And it's really focusing on what the concussion recovery profile looks like. So you might not need that extra level of the retina just to say yes, no concussion, but how do you ensure that safe return back to play and really monitor that the concussion has recovered? That's where we think some of the high accuracy retina work could get really exciting. And we correlated that with some of the gold standard work in the concussion space, looking at impact reaction time. We also did the VOM study, vestibular ocular motor test there, where a lot of our metrics correlated with nausea, which was very interesting from a symptomatic standpoint. And finally, what we have here, I talked to you a lot about what changes happen in fixation with disease, but on the flip side, if you think about fixation from a performance standpoint, what does it mean to be an elite athlete? And so we did have a study where we looked at eye motion changes in elite athletes versus the average bear. And so you can see that in the green here is just kind of the normative population, everyday you and me. And then in the blue and the yellow, we see kind of the minor and the major performers in the elite stage for baseball. And so what you notice is a clear shift in our stability. So BCA is a fancy way of just quantifying fixation stability. It's the bivariate contour ellipse area. And it's just saying my fixational stability as an elite athlete is much tighter than the fixation of the average person. And so that for us was really exciting, because could fixation be a way to probe performance? Could it be trained? Could it be a way to actually help improve performance? So these are areas that we've looked into all from a research standpoint and are really excited to explore further. And so with that, I will end here, just talking about, again, the value of fixational data. All the data I presented today was actually recorded with the research grade device, not our commercial device. So I will give the caveat of this, again, is research only information and does not represent the indications of use for the commercial unit. But it is exciting research that we as a company are looking to further. So eye motion changes for fixation, again, they can happen in a host of neurological conditions. And we're searching, can they be a biomarker for detection and monitoring? And maybe you can be the wonderful collaborators that help us do that. They also have the ability to potentially reveal localized regions of brain atrophy. So that's a study that we have right now in preparation. So I didn't share too many, any slides on that, but that's in the works. And then finally, looking at sports performance. How can we use eye motion as a way to not only look at disease, but also to look at the opposite spectrum of elite performance? So with that, this is me during my postdoc building the different devices I did to get the imaging that you saw today. Love to say that we're really at the intersection of industry, academia, and clinic, and I'd love to connect with all of you and learn more how we could potentially collaborate and partner. So thank you very much.
Thank you, Christy. This was a great talk. I can see already there are a lot of great questions here, and some of my colleagues in the other universities, they have already published on it. So I have Dr. Armstrong and Bijan. Can you guys talk about the paper you put in the chat?
So great talk. I really enjoyed that. Actually, the iSaccade, when you mentioned, it brought me back when I was postdoc, actually in Harvard. And I used to use iSaccade for assessment of motor learning, how we are learning a new skill, how we can model the pattern of forgetting of the new skills. That it seems that iSaccade at that time actually was a great application. But that work that actually David actually did it and can talk about it. And also now in my new role in UCLA as a research director of CASET, it's very important is how we can better evaluate the surgical proficiency in our surgeon. And that there was one earlier studies with genius idea of David that saying that how an advanced surgeon and novice surgeon can look at a radiography of, for example, a bunion, and actually the pattern that they look at it a radiography is different based on the skill that you're getting. I mean, some of the novice, David, do you want to elaborate on that research? That was very cool.
Oh yeah, sure. So I mean, this is now very old. This is now more than a decade ago, but this brought us back. This is again just a tour de force talk. Thank you. But so what we saw back then was that trainees, women and men, looking at a radiograph, would, if you looked at a heat map, kind of the saccades of them alighting on different parts of the radiograph, and this was just for a very basic kind of a radiograph for a bunion correction, and you could look at and they're to look at different areas, and they would look at one metatarsal, another metatarsal. They'd look back for this, what's called an intermetatarsal angle. And you saw them doing this. Then you switched over from a novice person looking at it to someone who was a master surgeon, and she or he would be looking at it, and they would just boom, go straight in on the first metatarsal phalangeal joint. And it was astonishingly obvious. And we thought that this could be a good way to train people, or to maybe gauge a clinician's kind of aptitude as they were kind of going through a lot of this as one other metric. But of course, this also has phenomenal applications for our patients at home. What are they alighting on in terms of eye tracking? And I'm sure you think about this all the time, but I'll shut up and let you talk about some of these things in terms of the applications, because there are myriad applications.
Oh my gosh, yeah. I mean, the eye is such a powerful place, and I hadn't even thought about surgical assessment, but that's really exciting. I mean,
Yeah.
the way in which we can assess really any task or job that requires high acuity movements and function is exciting. I think, yeah, I listed there, I think, just a couple of the disease use cases for neuro and mental health and injury and cardiac. I mean, the eye is just a beautiful non-invasive window into brain health. And so looking at surgical assessment, I wrote it down in my notes. I'm like, that's really cool. That also made me think of like pilots. How could you assess,
Sure, absolutely.
elite pilots? All of those kind of high acuity tasks got my brain my brain working.
Absolutely, positively. One of our staff used to train. She did a lot of the human factors design work for Lockheed in jets, and she was really fascinated with that some years ago. Bijan, that was Karen de Heiveter. Do you remember? And so you're absolutely right. Just no question
And
about it.
David, also you may know, and of course is a big deal for us. I share the CASIT Center, so that you can learn also about some of the potential application for surgical training. But one of the biggest challenge that we have is to identify cognitive fatigue among surgeon and the staff in the operation room.
Absolutely.
And
All of this. I mean, right now I'm wearing some Ray-Ban Metas, and these are just my daily drivers now for the last year. I don't even think about these things, but I have my, I could engage it just by asking it a question right now. But a lot of this also does have some ability that, the sensors in these may have some ultimate ability to measure saccades ultimately. Right now they don't, because there's no screen on there, but the next version will. But there's all kinds of stuff you could do now that this form factor is so much more just accepted. No one even thinks twice now. It doesn't look anywhere near as geeky and ridiculous as 10 years ago when we were wearing Google Glass. I have 2 pair of Google Glass in my drawer here. So
awesome.
all glitter brick, but it's a very different time.
Yeah, even thinking about potentially using the retina as a way to fine tune some of those glasses. I mean, that could be a really exciting thing to think about too.
And in diabetes, of course, many of our patients have proliferative retinopathy, but one can predict other complications. The old joke is stare into my eyes and you can see my soul, like my foot sole. And because when people get, and these are issues of doing, you can look for small vessel disease. There's quite a lot of data now where this basically proliferative retinopathy is collinear with foot disease, and you can do scans in a one-stop shop to be able to predict that now. And I could see that being something as well. So anyway, sorry to gangster up all the conversation, but I'll shut up now.
Thank you,
Thanks for your
David
insight.
and Tijan. So, Christy, I actually got to hear about you from Ryan and Enrique, our faculty members at BME, and the work you were doing with Paige Jones and our students, and I had discussions with David Biberdorf about your work. David, yeah, David is actually, I want to thank David for bringing speakers like you to us. And this is not the first time. A few times David has brought us really good speakers. David, do you have anything to comment?
No, Christy, you did a great job. This is kind of exciting stuff, and I can see how if your unit could be put into, let's say, extended care facilities or things like that, we can monitor much more closely things that could affect eye health for aging. I have tons of ideas I'll share with you offline here on some other things, but just to share with the group that Christy volunteered to use her instrument with our U.S. luge team, Olympic luge team, and try to gather data, because many of these sports like luge and skeleton and bobsled, they get done with a run and they have what's called sled head. And so what we're trying to do is see if we could use Christy's technology to measure subclinical concussion. So it's exciting, Christy, and I'm excited to thank you for sharing with us.
Yeah, absolutely. That was a really fun study for me. And I know, Ryan, we got to connect to go over the data a little bit deeper. But I did notice, with some of the metrics, looking at them longitudinally, there almost looked like it got worse throughout the course of the day, and then it would kind of recover the next day. And I'm interested to see how that might have correlated with accelerometer data to see kind of how that changed as well. And that was just one patient or subject, so maybe not everybody had that same trend, but it was looking really cool. So I'm excited. I think there's great publication potential with that as well, based on what we've seen on our side, for sure.
Ryan?
Yeah, I would just add, it was a real pleasure collaborating with Christy on that data collection. And we did use both Christy's Retinatrack as well as a balance platform. And we're really glad we had both devices, because when you're working with elite athletes whose muscles, they have very good control of their muscles. So that balance platform, that's we didn't get great sensitivity out of that, because the athletes are really good at controlling their muscles in their body. So being able to kind of bypass all those muscle systems and use Christy's device, I think, is going to give us a lot better sensitivity.
That's great. Thank you guys for all the input. Christy Jean is also there. You had something you put about the technology that your son is using. Christy, are you there?
Yeah, I'm here. I think it's this has been brought up on a previous talk. We have a lot of children in schools that struggle with reading. And my son tested off the charts in logic, and he was in the bottom 10% for reading. And it appears in third grade, because that's when you get the longer word problems. There's more reading going on, and his eyes were messed up. And our educators have no clue how the eyes and the brains work together. And so it's only because my sister's an optometrist that I even learned about vision therapy. There was no one supporting this kid, because he was just going to get put on special ed. And if you can actually put data to this, we could change. There's a number of kids that go through and experience this to varying degrees. So, you talk about diseases, there's just basic functioning. And then the other side of it is also now with long COVID. And a doctor had commented that long COVID is almost like a chemical concussion, like the brain can get affected. And depending on how your eyes and your brain are communicating, my sister has been going through vertigo for 2 years now, and they are doing therapy with her eye movements to the brain because there's just that disconnect. So if you can actually put data to it and be able to help track progress, that would be that would give hope to these people, because sometimes they don't see the effects of like the vertigo or what have you. But if they can have that data behind it that says, hey, your therapy is working, we have data behind your eye movements, you're tracking better, it just can make the world of difference when you can put data and science behind some of these thoughts.
Absolutely. I wholeheartedly agree with that. And I know, a lot of children that have reading difficulties or dyslexia, some of it is very dominated by tracking errors. I mean, if their eye is unstable and it's jumping to different letters, it makes it very difficult to be able to read properly. And so having vision therapy, having the capability to, one, detect that it's even happening, to your point, and then two, to have therapy that can potentially help mitigate it. A lot of the time, vision therapy, because the results have tended to be kind of subjective of, oh, wow, I feel better, I'm reading better. It hasn't been given as much clout, I think, as it deserves. Whereas if you have a technology like ours, you can do a before and after. You can show, here's how the eye was functioning in its pre kind of baseline state, and then here's how improved or different it is after VT. So I think, this can help not only identify children or identify difficulties, but also help the therapists in this space to show, the work they're doing has objective, not just subjective, has objective results at the end, which is really exciting.
Absolutely. So keep it up. Very exciting.
Thank you very much, Christy. We have actually Cheyenne having a couple of questions there, and his experience with Apple. Cheyenne, do you want to talk about it?
Yes, thank you. I don't know if it was posted to voice. That was fascinating, Christy. Thank you very much. Almost a year ago in May, Apple also introduced an eye tracking technology. A lot of people jumped on it to look into it for disability conditions or other applications. I was wondering if you have looked into it, and what is the gap there? Definitely it wouldn't be as accurate, as precise as the technology you built, but I was thinking, can it be used to some degrees for detect some early signs of Alzheimer or other conditions that can lead to physicians using your technology for that? I was just curious about at what degree that technology works compared to what you have built or know about. Thank you.
Yeah, so I think, tracking eye motion at any level, I think, is going to be valuable. It just really depends on kind of what state you're coming in, how kind of pronounced the eye motion abnormalities are when they get detected. So typically when you have kind of the pupil tracking or the anterior eye of the being tracked by your phone or your webcam or your glasses, those are going to be a little bit less accurate. They're probably going to see, again, down to 2 degrees, down to 1 degree at most. And so you're not going to see potentially the subtle changes. So like the idea for early detection, early intervention, subtle monitoring, that can't happen. But, if a movement is large enough to be seen at bedside, a phone can certainly see that as well. And so if you have more, for example, rural individuals who don't have as much, availability to get to a clinic, something that a doctor could potentially see by the naked eye at bedside, a phone could potentially, build up the algorithmic power to help. So I think there's so much potential in what, the eye can offer to the space. And I think where I find that exciting for us to potentially, plug in the retina is, how can the retina help to make the pupil, help to make kind of the stuff from Apple or, the phone-based stuff? How can we help to make and tune it to be just as good as retina? How can we begin to create, ML and AI that can really assess at a fine level what we do and then transfer it over to another methodology? I think that's going to be, for me, the most exciting part, I think, that we could offer to this space that does a lot of the pupil tracking. But, we're looking at movement down to 30 microns, 25 microns, so really, really tiny. Typically, a degree of the retina is about 300 microns. So again, you'll see changes. They're not going to be as subtle as the ones we'd pick up. So the idea for early interaction might be hard, but certainly at some level, it could totally pick up, larger changes.
Thank you very much. That was fascinating. Thank you.
We are on the hour, so if anybody wants, but if Chrissy, if you have a few more minutes, I'm thinking, Bijan, how do you see collaborating with CELI in context of C2SHiP?
Wow, there are a lot of applications. As you mentioned, definitely we are looking for maybe some joint collaboration on SPR, for instance, for dementia is a huge problem, as well as cancer-related fatigue. These are the two important topics that I know that NIH are looking for, having a better digital biomarkers to better characterize those conditions and severity. Definitely we can run a variety of the clinical study, Christy, for you, just if you are interested to examine that, let's say that in operator theater for surgical proficiency, how that system can identify the fitness of surgeon to do surgery, because currently we don't have a good tool and many surgical mistake is happening since we don't have a better tool to evaluate those kind of fatigue, as well as the other area, in area of the dementia, in area of the cancer, management of cancer. These are the areas that we can help you. Management of cancer, I emphasize on this because now there are a lot of efforts on actually improving patient-reported outcomes and better evaluating the side effect of the chemotherapy and radiotherapy, and cancer-related fatigue is a huge problem. There's some good programs happening at MD Anderson that trying to address cancer-related fatigue to boost the outcomes. And I believe that that's one of the areas that you want to explore as well. But Dr. Armstrong, I believe that you are doing a lot on this area, as well as Dr. Armstrong mentioned, diagnosis of neuropathy and as association complication with diabetes is a huge problem. I know that some eyes tracking currently has become very popular to look at early sign of neuropathy and also complication that patient may have. If we can having very quick tests that evaluate those kind of conditions, that would be a great value as well.
Absolutely. Yeah, I'm super excited to develop, further collaborations and partnerships. So by all means, anyone that didn't get a chance to ask a question, please ping me offline, connect on LinkedIn. Happy to talk about, anything fun scientific that we could do together.
Christy, I'm hoping that you can come to UCLA. You're in San Francisco, I believe. Am I right?
Yes. So we have a warehouse in Santa Clara. I'm there right now.
Okay. So I'm hoping that you can visit us in Los Angeles and attending our semi-annual meeting at C2SHiP in UCLA. So that would be great also to meet with several clinicians and surgeons, as well as other industries that might be interested in partnerships. So you're more than welcome to join our semi-annual meeting. And to everyone else, please don't forget to register in this meeting. That would be a great opportunity. There is a plan to give a tour to CASIT facility and simulation surgery at UCLA. Is one of the very first in Southern California. David, sorry, I know that USC is great, but UCLA is better on this area. But hopefully you can see the latest innovation in this area. I know that Intuitive Leadership, they plan to attend, and they might be interested about some of the work that you're doing on actually eyes tracking as well.
Yeah, Michael, I have two questions in mind. Do you want to ask it? I don't know if you have, Christy, a few more minutes for the couple of questions left.
Sure, I got a couple of minutes. I'm in a hotel right now, so I have to check out by 10. So I probably have time for like 2 more questions.
Okay, Michael.
Oh, my question really doesn't deserve that much. Thank you very much for the talk. Whenever I hear about new biomarker modality, I get excited because there is so much to explore yet in this new types of biomarkers. I was just curious about a pretty benign topic of getting tired out of the screen time. So how can we use maybe your device to evaluate the quality of the screen or general tiredness of people when they spend time in front of the screens, computer screens. Maybe in terms of formal and informal research, just curious if you did any kind of thinking in this direction.
We haven't done anything with screen fatigue yet. The fatigue that we did was really kind of focused on, we did the multi-dimensional assessment for fatigue. That's the kind of survey that we had given out for the MS study to controls and to MS patients. And that particular one, the controls didn't have any correlation with our fixational data, only the MS patients did. But I do think depending on the survey, you might elicit different responses. So if a survey was particular to more visual aspects, that survey had a lot of like functional motor movement questions and not focused just on vision. So I know that there are visual fatigue surveys in particular. Maybe there's even some, I'm not sure, for screen fatigue as well. So I think if we get very pointed with how we ask the questions and how we see kind of teasing out more specific fatigue, is it screen-based, is it motor function, with ambulation, those differences, I think depending on the survey, we could absolutely kind of start to tease out some of those differences. So that could be cool.
Thank you.
Thank you, Michael. And Marge, do you want to go for the last question?
Oh, thank you. I just was wondering if there were any effects on eye movement based on, for instance, age alone. You mean, does our eye movement change just because of age? Or are there differences between men and women or ethnicity? Does that play into it? And the other part of the question had to do with, nearsightedness versus farsightedness, or the kind of job that somebody does. And I know you mentioned about pilots, or I was also thinking, as Michael was talking about, people doing close work on the computer screens, what difference that might play into it too.
Yeah, so for your question, I have a slide on that in case somebody asked about age. So we do actually see changes with just normal aging. So at UCSF, we have the privilege of utilizing a cohort there where they're doing cognitive testing as well as PET scans to ensure that they truly are, quote, healthy, normal aging. And so that allowed us to start to look at and we broke it based on over 50, under 50, just so the N would be roughly the same. And so I think there's a lot more we could do here with like when particular changes happen. But what we found is that with the microsaccadic metrics for age, there was statistical difference between the under 50 versus the over 50 group. And it was looking at particularly amplitude and peak velocity and acceleration of microsaccades alone. So those are some of the things that we've started to tease out. And again, they overlap quite a bit, but there is kind of a statistical difference, at least, between the magnitude of the velocity, mostly in the horizontal component, as well as the magnitude or the amplitude, again, mostly in the horizontal component. Generally, human vision is really along that horizontal meridian. It's once you kind of get into these neurodiseases that you see a lot more of the X component being dominant. So that's why, the X or the Y component is generally low for kind of the norms. But it's very interesting to me to kind of have this baseline of, okay, the natural aging process has these changes in microsaccades. And then neurodegeneration basically just speeding that up, right? Having that happen within a shorter time frame based on the disease. So we looked at acceleration as well. Acceleration, those are all linked based on different derivatives, right? Amplitude, velocity, acceleration. So they're all linked through the microsaccade world. And then, for us, this allows us to have a more well-rounded comparator. So knowing that you're going to see changes in normal population, you can then account for those when you're actually looking at neurodegenerative disease as well. For the nearsighted and farsighted, that's a really interesting question too, because there's a researcher out of University of Rochester, Michaela Rucci. He focuses a lot on myopia and the changes in fixation that could occur with myopia. And interestingly, microsaccades aren't the focus of the change with myopia, it's actually drift. So if you check out Michaela Rucci, University of Rochester, they have some really interesting papers looking at eye length and how the drift characteristics of the eye change according to that myopia level. We haven't done a lot of work yet looking at drift, but that'll definitely be interested in and something we'll have to take into account when we do go kind of the drift analytic route.
Okay, great. Thank you.
Thank you very much, Christy, and staying longer. This was a great talk. We really appreciate it and looking forward to collaborate with you in some ways. We'll figure it out.
Yeah. Hey, by the way, Kuya, is there any way to get the tech that those papers that I put in or those posts that I put into the chat to her? Just cut and paste them. I'm sorry, I just.
You know what? I will send an email to her, copy
Good.
you on it, David, so you can make the connections there. But I will send it.
And I was also connecting her with my friend Mike Conti, who's the head of vascular. He's texting right now, all asking about this he has a big interest in limb preservation too, but to be continued.
Sure.
So we started looking at blood flow. I didn't show any of that today. We started looking at blood flow though with our videos. So we're starting to look at speed of
I'm
flow.
telling you, that is a big area. I mean, there's a tremendous amount of these different matrices are very, very similar here and then at the end of the body and also in the kidney. So all this microvascular disease. So to be continued.
Thank you all. Have a great weekend.
Thanks,
Bye-bye.
everyone.